Okay, so thank you so much for the opportunity to come and speak here today. So today I'm going to tell you about Blessy, which is a new tool from my research group. So of course, genes in the genome are transcribed into RNA, which can then be translated into proteins. And we know that in the human genome, there's roughly 20,000 genes that can code for proteins, but in fact, there's many more proteins that can actually be produced. And the reason for this is that genes can undergo alternative splicing. And so we can have numerous isoforms or transcripts for each gene. And of course, then we can end up with multiple different proteins that are produced. These proteins can actually have quite different function. In fact, they can even be opposite. And a really fantastic example of that is Drosophila, where there's a key gene that actually determines sex, which when it undergoes certain splicing and results in a cascade of events that gives you either a male or a female. Now, long reads are a really exceptional way to actually understand splicing and to look at transcripts and their abundance. And we know that it really overcomes these limitations that we have with short read sequencing with the fragmentation. So essentially we can link, or what I call phase, the events at one end of the transcript with those at the other. So how we might actually analyse this in practice is that we can take our long reads and we can map them to our reference genome. We can then say how many reads do we have that are consistent with each of our different isoforms, and we can build what we call a transcript count matrix. So we do this for every single sample, and hopefully we have replicates as well. We have a good design. And then we can ask, do we see a statistically significant difference in the proportion of a particular transcript relative to all the other transcripts in that gene? So when we do this in practice, we often end up with hundreds of different genes that might have this differential usage occurring. And so we have to try and make some sense of that. And so we can do that with some functional analysis. And often what we'll do is we'll go and actually look at what proteins are produced. We'll look at the sequence of them, the open reading frame. But to try and determine what those functional differences are, we might look at something called protein domains. So these are regions of genomes that are somewhat conserved. They tend to fold independently, and they have quite distinct functions. So an example might be a region of a protein that actually binds to another molecule like DNA. And if we see that those regions are spliced in or out, we can then maybe infer that that might impact the actual protein's function. So some good examples of this come from cancer, where certain regions that are removed actually result in that protein being a dominant negative and blocking the function of the other proteins and actually giving you a really opposite effect. And so in our group, we thought, well, if what we do in the end is actually try to interpret these changes through these protein coding domains, maybe we could actually take this step forward and we can actually sort of directly try and do our statistical testing on this feature instead. And so what we propose to do is to actually map protein domain regions to transcripts, and then to make the assumption that different combinations of protein domains in our transcripts, if we see the same combination, that they might be behaving functionally similarly. And so we can group our transcripts in this way, and we can generate what we call a domain combination or a DOCO count table. And this has a couple of advantages. So one thing is that it's more sort of directly interpretable. It's taken away some of that manual process of trying to understand what the changes are at the protein level. But actually the other advantage is that we now have fewer features. So we have this level of resolution that sort of sits between genes and transcripts, but hopefully retains the important biological information. And so because we have fewer features, we may have a higher number of average counts per feature, giving us more statistical power to see differences. So in terms of practical implementation of how you might do this, I have a master's student in my group, Liam Lee, who's built this software package in R called Blessy. And what it will actually do is it'll take a number of protein domains from USC genome browser, and they have a number of different types of domains that have been annotated there, and it will map those to transcripts and give you a list of all of the domains that each transcript has. And then it can do a second thing, where it takes a count table of transcripts and it will actually aggregate them to give you a DOCO table, which can then slot into all of the regular statistical testing packages that we like to use, things like EDGAR, LIMA, and DEXSeq. Okay, so to test whether this idea actually works and to test that our software is doing what we think it does, we did a little bit of simulation. I won't go through all the details, but I'll just briefly mention that what we did was we took 212 genes and we randomly selected, and then we took 2 transcripts in each of those that had different domain combinations, and we just switched their expression between conditions, and we did that across a range of coverage levels. And we compared the standard approach, which uses transcripts, to our doco approach with Blessy. And these are the results here, showing you across different coverage levels. And in the green, you can see the docos compared to the blue in transcripts. So we pick up more true positives and fewer false positives. So a nice validation that the method works. But the area where we're really interested in applying this is, in fact, is in long-read single-cell sequencing, which is a really absolutely fantastic application of long-read nanopore, and it's been using more and more in combination with 10x genomics. And so one of the challenges with actually trying to do things like differential transcript usage in long-read single-cell analysis is that you end up with a really enormous count matrix. It's very, very sparse. So you could have, for example, 380,000 rows of transcripts and tens of thousands of columns, which are your cells. And so if we can try and aggregate this data to make it a little bit less sparse, hopefully we have some increased sensitivity. And so again, we did a little bit of simulation to try and check this. And I won't go into the details, but just to show you, this is the area under the ROC curve. And we tested a range of coverage levels as well as numbers of cells. And you can see the doco is that green line, and then using regular sort of transcript approach is in the purple. And so you can see we have a higher accuracy, and in particular, when we have low counts. So in this really sort of more shallow sequencing scenario. So I just want to finish off by giving you a little bit of a practical example of how we might apply this in a long-read single-cell pipeline. And I'll do this using 2 different data sets. One's a public data set called SC Mixology from some colleagues of the WEHI. That's a mixture of 5 lung cancer cell lines. And then we also have a patient who has acute myeloid leukemia with a sample we've been analysing in my group. So the first thing you do when you have your 10x genomics long-read data and you've base called it is you actually have to get those 10x barcodes and UMI sequences. And we've developed a tool called Flexiplex to do that in my group. You can have a look at it. It's published a couple of years ago. The next thing we do is that we try to remove our PCR duplicates. And so we've just developed a new tool called Nail Polish, and there's actually an online poster about this, so you can have a look if you're interested. And what it does is it will take reads that have the same barcode, UMI, and it will actually collapse them and consensus call them to try and improve the error rate. And we find that up to 50% of reads can actually be PCR duplicates. So this can significantly improve the actual error quality, or the quality of the data. You can then align to the reference genome. You can quantify with ORFISH, or another tool that you're interested in. And then we can aggregate our transcript count table to get our domain combinations. So just to show you the difference between those, we find that we get less than half the number of doco features compared to the number of transcripts. And because of that, we have a much higher, really significantly higher number of min counts for our docos than our transcripts. So suggesting we may have more power in this scenario. Because this is just a count table that's been generated, we can pop it into whatever our favourite tools are that we would normally use for gene level expression or for transcript level expression. So for example, we can make a UMAP, or we can do clustering. These UMAPs have just been generated with Seurat. And what you can see here immediately is that they actually look really similar. Transcripts and docos look really similar. And so that really largest sources of variation are retained. We're not losing that information by aggregating our counts. And I'll just mention a little bit this bottom one, because I think this is quite interesting as well, a little bit of an aside, but you can see in this leukemia sample that the cells on the left are actually a healthy group, and then you see these sort of 2 branches, and this is the leukemia. This particular patient actually has a subclonal disease that has a mutation in the gene NPM1, and we could actually identify the cells that had that using Flexiplex with our long-read nanopore data. So quite a sort of an aside, but quite a nice kind of application of long-read sequencing. And they're shown in the red. So once we've done that, we can then do our statistical testing for differences in usage. And we use Isopod to do this. And the author of Isopod is actually here, has a poster in person, so please go and have a chat with him if you're interested in this. So we can compare our groups, and I won't give you any sort of specific examples, but I'll just show you the overall kind of numbers that we get when we do this. And so what's shown here are these plots are a little bit complicated, but basically it's showing the inverse of p-values for transcripts versus our DOCO method. So the top right-hand corner is actually the most significant genes. And the purple, which might be a little bit hard to see, is the transcripts versus the green, which is our DOCO. So essentially this tells us that around half of the genes where we pick up this sort of differential usage, they don't actually result in a change in domain usage at the protein level. We also pick up some events that are just seen using DOCOs, and they tend to be the ones that have the genes that have lower counts. So again, seems like we have higher sensitivity when we have more shallow sort of sequencing or lower counts. So I'll just finish there and say we've developed this new method. If you're interested in trying it, please have a go, and feedback's very welcome. It's also worth mentioning that you don't have to use protein coding domains, so you can actually provide it with any type of annotation. So you could do, for example, differential exon usage if you give it the exon annotation or whatever regions of interest you're interested in. So with that, I'd just like to thank everybody here for their contribution. In particular, acknowledge Liam Lee, who was the author of the software that I presented today. So thank you.